In this video, we're going to be talking about nucleic acid-based biosensors. To begin with, here is the suggested reading. I have already posted these papers and this chapter on the track site, but basically the first chapter is from the same book that I had already mentioned that you have access to from the library for all of the chapters. In addition to that, there's two review papers. The one that I like the most is going to be this paper that very clearly discusses the area of nucleic acid-based probes. Also, as a disclaimer again, a lot of these notes were actually initially developed by Dr. Shannon Wygum, and I have modified them over the years. Nucleic acids consist of two main types in nature, DNA and RNA. DNA stands for deoxyribonucleic acid, RNA stands for ribonucleic acid. And the difference between these two, if we look here at the right-hand side, which is the structure of these nucleic acids, is in this X. In RNA, the X is actually a hydroxyl group, while in DNA, we're going to not have that hydroxyl group, and instead, what we're going to have is a hydrogen here. So that's the main difference between them. In addition to that, there's going to be some difference in the bases that we have from down here, but I'll get to that in just a second. Now, the nucleotides that are basically the monomeric units of your polynucleic acid here are going to consist of nucleotides, and those nucleotides have three main components: a nitrogen base, a 5-carbon sugar, and phosphate group. And we can see that here. So what's here in this dashed circle, that's what's known as the base, and there are several types of bases that are described down here. In addition to that, we have the 5-carbon sugar shown here. And again, that's going to be a little bit different for DNA versus RNA. And then finally, we have the phosphate group that is going to be found in between nucleotides, kind of linking different nucleotides together. Now, some additional things that I want to point out while we are in this figure is that there's a specific labeling order for the carbons in the sugar. As you can see here is label 1, 2, 3, 4, 5 for that sugar. And that might be something that is kind of important for you guys to remember, because once we go to actually form a chain of multiple nucleotides, what's going to happen is that the linkage is going to be between the 3' and the 5' end of the sugar. So from here, of course, we're going to a phosphate. That phosphate will link to another nucleotide. But then from 3' here, we go to this oxygen, which will bind to another phosphate from another nucleotide. So that's just something to keep in mind. Now, coming back to the bases down here, if you look at them, you can see that there are some similarities between them. We're going to have two main types of bases. Some of them are called pyridines. Those pyridines are going to be, let me see if I can highlight them with a specific color. Let's say pyridines are going to be this guy, and this guy. As you can see, they basically have this pyridine structure with some differences in terms of methyl groups, for example, that can be found in some of them, or some of the double bonds that you might find in the cyclic structure. In addition to that, we're going to have purines. And these purines are going to be structures that contain a pyrimidine ring as well as an imidazole ring that is linked together. And of course, those are the remaining ones, basically. So this guy, and this guy are going to be your purines. Now, in terms of which of these are found in what kind of nucleic acid. So adenine, guanine, cytosine, and thymine are found in DNA. Adenine, guanine, cytosine, and uracil are going to be found in RNA. And then finally, this hypoxanthine might be something that you might not have ever heard about. It is a natural nucleic acid base, and it is occasionally found in transfer RNA. We're not going to really be talking about that one too much at all. Let's see. There's one additional thing that I wanted to mention while I am here, And actually, two things. One of them is that nucleic acids tend to be a little bit unstable in the sense that they can be cleaved by nucleases or enzymes that are designed to cleave nucleic acids in your body. And so the use of these nucleic acids for an in vivo application might be a little bit difficult because you might have cleavage of that nucleic acid before it has the ability to interact with the target molecule of interest. However, there have been many technologies that have been utilized to be able to improve the nuclease resistance of these nucleic acids. And that's basically by doing chemical modifications on the structure. Some of the chemical modifications that have been published or that are readily utilized even by some companies is the modification of this X. So modifications by changing the X to a fluoro, like a fluorine atom, or changing it to a methoxy group, which is an oxygen bound to a CH3. That has been demonstrated to increase the stability. Another modification that can be done is to replace one of the oxygens of this phosphate with sulfur to generate what is known as a phosphorodioate molecule. And again, those basically increase the stability by preventing the enzymes from actually acting upon the nucleic acid. The last thing that I wanted to mention before I moved on from this slide is that there are some synthetic nucleic acid based structures. So they're going to be structures that are different from the chemical structure of DNA and RNA. And the two that I wanted to mention here are going to be peptide nucleic acids. And a peptide nucleic acid is going to be one that is made through peptide linkages, very similar to those that make up proteins and peptides. So the backbone of the peptide nucleic acid is going to have the structure of nitrogen to carbon, where this is going to be a carbonyl basically. And then from there repeats on and on and on. So nitrogen, carbon, carbon, with a carbonyl, nitrogen, carbon, carbon, etc., etc. Now, in these peptide nucleic acids, what we're going to have is that the bases that provided with nucleic acid type of structure are going to actually be bound to the nitrogen. So from the nitrogen, we're going to have some of these bases, sometimes with the sugar, sometimes not with the sugar, being attached. And so all that we're doing is really changing the backbone, but still providing the bases, which are the ones that are required for hydrogen bonding to take place. The other type of structure that I wanted to mention are click nucleic acids. And these click nucleic acids are being developed by the group of Chris Bowman at the University of Colorado Boulder. And basically what happens is that in these cases, we're utilizing click chemistry, specifically diol-in chemistry, to be able to develop a backbone that is going to be able to present these bases and enable hydrogen bonding to take place. That's still under development. They have some papers showing the feasibility of that technique, but they still have a ways to go to make this suitable for biosensors. All right, in terms of the structure of nucleic acids, the nucleotides are going to be linked through phosphate residues at the 3' and 5' or 3' and 5' carbons of the sugar moiety, as I had already mentioned. So basically, if we look at our sugar here, number 1, number 2, number 3 carbon, number 4, and number 5 carbon. As we can see, we have linkage to the phosphate group from the 5', and then we have linkage to the phosphate group of the next nucleotide on the 3'. The second thing is that the direction of the chain of your nucleic acid is recorded from the 5' to the 3'. That means that if you are going to be maybe buying some nucleic acid and you're going to be listing the structure of that nucleic acid, you're going to list the nucleotides by utilizing the sequence from the 5' end and going down. So in this case, it would be A, G, C, T, basically. And that's shown here. And the typical notation is to write 5' on one end and write 3' on the other end, just to indicate that that is the case. Now, another thing to keep in mind is that you have all of these phosphate groups along the backbone. Those phosphate groups are negatively charged, as you can see here. And so because of that, your nucleic acids are going to be negatively charged overall, and they can be considered polyanions. As you guys know, if you have two polyanions, they are actually going to repel one another due to charge repulsion, right? So what's important is that these nucleic acids cannot come together to form what we know as the double-stranded systems unless cations are present, such as sodium and magnesium cations, that are going to stabilize the structure and enable them to come together. So that's just something to keep in mind, that in any kind of biosensor system that you might be utilizing, you actually need to include cations as part of your buffer. So a lot of salts, and these divalent salts tend to be like really, really important for the function of these nucleic acids. All right. The next thing that I wanted to mention is that, as you guys already probably know, is that nucleic acids can form complexes with one another, specifically two polynucleotide chains that have a complementary sequence can actually come together in what is known as a process of hybridization. So hybridization is the formation of that double-stranded duplex kind of thing. And this hybridization occurs through hydrogen bonding between the complementary bases. Specifically, adenine binds to thymine, cytosine binds to guanine. These pairs are known as Watson-Crick pairs. That's kind of the normal way of hybridization. However, I'll go ahead and mention that there are some non-canonical hybridizations that could occur that we're not going to mention right now. This right here can happen in between two completely separate strands, as is shown here. We have one from the 5' to the 3' and from the 5' to the 3'. Or it could happen between complementary portions of a single polynucleotide that basically folds upon itself and has complementarity. The other thing that I want to mention is that these are anti-parallel, and we can see that here. We go in one direction for one of the strands, in the other direction for the other strand. Now, a single polynucleotide strand can fold into a specific conformation that is stabilized by intramolecular hydrogen bonds, and that's known as a hairpin configuration. What I mean by that is that you could have a polynucleotide, something like this, but if there's some complementarity between this end and this end, for example, that nucleic acid can fold into something like this, where now we're going to have hybridization between those ends that are complementary. And that right there is known as a hairpin conformation because of the shape that we end up with. Now, one thing that I forgot to mention when we were here is that the different pairs actually have different numbers of hydrogen bonds. So whenever you have A binding to T, you actually have 2 hydrogen bonds in between them. When you have C binding to G, you have 3 hydrogen bonds that are bonded to one another. So C to G is 3 hydrogen bonds. I'm not saying that this is a double bond and a triple bond. What I mean is 2 hydrogen bonds and 3 hydrogen bonds. And you can see that in the structure here. So between A and T, you can see the 2 hydrogen bonds occurring. Between C and G, you can see 3 hydrogen bonds. Now, for the case of RNA, remember that the T is replaced with U, and so what you have is A would bond through 2 hydrogen bonds to uracil. All right, the last thing here is that whenever the DNA ends up folding or something like that, like let's say to form a hairpin structure, that tertiary structure is going to enable binding of that nucleic acid to a particular analyte via shape-fitting and non-covalent interactions, in the same way that an antibody had the ability to bind to the target analyte simply by the proper fit and the proper electrostatic interactions, right? So when I talk about tertiary structure, maybe I should go back. The primary structure is the sequence of the nucleic acid, something like, in this case, A, C, T, G. The secondary structure of your nucleic acid is going to be once it actually kind of hydrogen bonds to its complement, either whether that complement is due to binding to a completely separate strand or binding to itself in a hairpin conformation. The tertiary structure is then the geometry that it forms upon folding or upon binding. And as you can see that here on the right hand side, this is a hairpin configuration, but you can see that the geometry is not just like a straight hairpin as what I drew here, right? There's going to be a three-dimensional structure to it. And that tertiary structure is what provides the right fit and the right presentation of functional groups to enable binding to a particular target. And that's something that is going to be important for biosensing because the nucleic acid needs to be able to bind selectively to a target molecule. All right, continuing on nucleic acid structure and properties. So when DNA duplexes are subjected to extreme temperatures or pH, the hydrogen bonds can be overcome, they can be cleaved, and the two strands separate. And when I say two strands, it could be like two completely different strands, or it could be the hairpin configuration that I mentioned before, where a single nucleic acid has folded upon itself. Whenever you break these hydrogen bonds that are keeping the strands together, that is known as denaturation. So notice that it has some similarities with the denaturation of a protein, but at the same time it's a little bit different. In the case of a protein, when you talk about denaturation, you're talking about the whole geometry of that molecule changing and thereby the protein becoming not active anymore. In the case of a nucleic acid, you are breaking the hydrogen bonds, and so the shape is also going to change. However, that nucleic acid actually can come back, and that's what I'm talking about here, that denaturation is reversible. So that the difference from a protein is that your nucleic acid is still active. It can still be reactivated. And so it's not like denaturation means it doesn't mean that that's the end of that nucleic acid for a sensing purpose. All right, so when denaturation occurs through heat, the DNA is said to melt. So it's not the same kind of melting that you or I think about when ice melts. It's just that we're breaking apart those hydrogen bonds, and so the shape of the nucleic acid is going to change. This process is going to occur at what's known as the melting temperature, or TM, which is defined as the temperature at which 50% of the DNA is present in its duplex form, duplex or hybridized form. It is reversible, and the denaturation and the renaturation or rehybridization occur in both double-stranded polynucleotides as well as in single-stranded duplexes. And this is important because it enables us some flexibility in the use of nucleic acids for biosensor applications, where we might be able to utilize just a single strand, or we might utilize two strands to be able to give us the signal. And you'll see examples about that in the later slides. All right, the melting temperature of your nucleic acid, which is the temperature at which 50% of the oligonucleotides are hybridized, is going to depend on the following factors. The first of them is the length and the sequence of the DNA or RNA. When I say length, it means how many nucleotides do you have in your polynucleic acid. The higher the number of nucleotides, so the higher the length, it's not letting me write. Let's try. Okay, the higher the length, the higher the melting point. And the reason for that is that if you have a longer sequence, let's say A, T, G, C, 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 A, T, T, A, once it folds or once it binds to its complement, then you're going to have more hydrogen bonds, right? If I'm only binding to 3 nucleic acids, let's say AAT, then I have 2 hydrogen bonds here, 2 hydrogen bonds here, 2 hydrogen bonds here. So there's less hydrogen bonds for me to melt, for me to disrupt. While if I'm hybridizing to a longer strand, let's say here T and here G, 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 oops, that's supposed to be a G. All right, let's try. All right, anyways, you guys know that I'm trying to draw G's. And then, so you would have more hydrogen bondings that would need to be overcome. And of course, remember that between G's and C's, you have 3 hydrogen bonds. And so the more hydrogen bonds that you have, the more energy you have to provide to disrupt them. And of course, because of that, your melting temperature is going to increase. When I say sequence, I mean what's the percentage of GCs that you have versus ATs? Because the higher the GC content, the higher the number of hydrogen bonds that nucleic acid can have against its complement. So the higher the GC content, the higher the melting temperature. The next thing is cation concentration, meaning in your solution, in your buffer system, how much of those sodium, calcium, magnesium ions do you have available? Those cations have the role of stabilizing the charges of the nucleic acids so the nucleic acids can actually come together. And typically what we're going to have is that the higher the concentration of those cations, the more stability you're going to have in terms of the hybridization, and the higher the melting point. In terms of concentration, I should have used a better word. What I meant here is the concentration of the nucleic acids themselves. The higher the concentration, the higher the melting point. And that's something that is a little bit interesting, but basically the more concentration of nucleic acids that you have, the more stable they're actually going to be. And that's something that is found experimentally. And then last thing, if you have any kind of denaturants, any kind of chemicals that are able to disrupt hydrogen bonding in between the strands, such as dimethyl sulfoxide, then of course that's going to actually decrease the melting point. So that's the only thing that is going to be decreasing the melting point, the presence of denaturants. All right, so how does one get nucleic acids for working with as a biomaterial or as a biosensor? Of course, you could obtain a natural DNA. DNA is present in pretty much any cell of any organism. So you could obtain it in that way. But of course, if you do that, then you don't have control over the sequence and you don't have control over the functional groups that you might need to attach it. So what's typically done is that we can purchase synthetic DNA that is prepared through solid phase synthesis. In that solid phase synthesis, what happens is that you begin with a solid support, which is typically a controlled pore glass or a macroporous polystyrene. And we can think about it as something like this, like I'm going to have a polystyrene bead. And on that polystyrene bead, I'm actually going to covalently attach my first nucleotide. Let's say that I'm going to attach my A. The chemistry here has to be special for me to be able to cleave it at the end. But after I do this, I can imagine that I have a lot of these beads that are going to have a lot of these sequences starting on their surface. And all of these beads are immobilized in, think about it like a tube. So they're stuck in a tube, but I can pass, and these are large, like micrometer sized. But through the tube, I can flow small reagents, like small molecules, like nucleotides, for example. And so what I can do is that I can add my first nucleotide, which is A, right? And then I can wash with buffer to remove any A that hasn't bound. After that, I'm going to add some of my next nucleotide, which let's say it's a T. And there's going to be a chemical reaction that now is going to put T on all of my A's that I initially had on the polystyrene beads. And after that, I wash with buffer and I remove any unbound T. I then add my third nucleotide, which let's say it's a C, and so on and so forth. I continue washing and attaching, washing and attaching. And so in doing this, you're able to control the sequence that gets added. At the very end, after you have synthesized the sequence that you are interested in, what you're going to do is that you're going to release the nucleic acid by cleaving the bond to the support. So as I mentioned, this chemistry right here is such that I can cleave it by changing some of my conditions or adding some kind of reactant, and that's going to basically let go of that nucleotide so that I can collect it. One thing that is really important about this is that we have not only sequence specificity, but we can also do chemistry to put fluorescent labels or to put quenchers or to attach linkage like functional groups that I can utilize for linking my nucleic acid to some kind of surface. For example, in this case, we're thinking about biosensors, so we're going to have to immobilize those nucleic acids to a surface, to our transduction system, and so we need to be able to have functional groups that are going to enable us to do that. On the right-hand side, I'm showing some of the commercially available linkage or functional groups that can be attached to nucleic acids. And so one of them is primary amines, and this is actually how the primary amine would be attached to your nucleic acid. So on the 3', for example, where the phosphate group is, they're going to be attaching this 6 carbon chain that has an amine group. This particular one actually can be attached either a 3', 5', or they also have internal primary amines. Similarly, we can attach this molecule, which the important part of it is this sulfur bonds. This right here will actually be cleaved later on by a chemical reaction to provide you with an SH, a thiol group, which you could then utilize to be able to attach the nucleic acid to a gold surface, for example, or a metal surface. Another functionalization that you can do of your nucleic acid is to attach biotin to it, or alkyne groups for click chemistry. And let me back up. Remember that biotin can be utilized to be able to bind to avidin or streptavidin. Alkynes are going to react with azide groups in click chemistry to form a triazole bond. And the last thing that I have here is what it's known as an acridite group, and the main part of it is actually this double bond, which is something that would enable you to now polymerize the nucleic acid as part of a free radical polymerization that uses those vinyl groups, those CH2-CH2 bonds that we have mentioned before. And anyways, all or most of these functional groups can be placed at the 3', at the 5', or internally, so you have site selectivity as to where you want to put this. One of the main companies that you can obtain DNA from is going to be Integrated DNA Technologies. I have worked with a couple of companies and I have found that they are the most reliable. This is their website, and one cool thing that they have and that I hope that you guys take a look at is Oligoanalyzer. So this is a tool that actually provides you a lot of information. If you just type in the sequence that you are interested in, it's going to analyze it to provide you with the estimated melting temperature based on the sequence. It's going to give you the molecular weight, the estimated extinction coefficient. Is going to tell you what kind of hairpin structures could it fold into, and how stable are those hairpin structures. So for example, here when I put in a particular sequence, it told me that that DNA could actually kind of fold. It's going to have a hairpin here, and the end of it is going to hybridize with this middle part of the DNA. And each of these red and blue dots indicate base pairing between H's and T's and G's and C's. It can also tell you about self-dimer formation, meaning that if you have two nucleic acid sequences, do they have the potential to hybridize with one another? So it's two of the same nucleic acid sequences. Can they hybridize with one another? That's something that you typically don't want to have. And then it can also tell you about heterodimers, meaning if you put two sequences, how well are they going to match, or how complementary they are, and how stable is that complementarity, what is the melting temperature of that complement, etc. Other companies that, well, there are many other companies that can also do this solid phase synthesis of nucleic acids, but one of the other ones that I would recommend is Eurofins, which is associated with Fisher Scientific or Thermo Fisher Scientific. All right. So the next few slides are going to be talking about platforms that you can utilize in order to detect nucleic acids, meaning that we're going to have a biosensor where the biorecognition molecule is an oligonucleotide or a nucleic acid chain, but what we're trying to detect is also a nucleic acid. For example, for the case of detection of a virus, we might want to actually detect the particular nucleic acid that is found in that virus. So what we're going to do is to try to detect that particular DNA, and we're going to do that through the interaction between the two nucleic acid sequences. One is the biorecognition molecule that we have immobilized onto our transduction surface, and one is actually the sample that is coming and that we want to detect. So basically, again, what you would do, it would be to immobilize the probe through a range of different chemistries, depending on what your substrate is down here. Adsorption is actually not something that is commonly used for nucleic acids, unlike proteins, for which for which that actually seems to work okay. So typically, you're going to be doing chemistry to attach the nucleic acid. From there, in this particular example, what we're doing is that we're actually labeling the nucleic acid, for example, with a fluorescent molecule. And if that nucleic acid comes and finds its complete complement, then it's going to stick there and it's going to provide us fluorescence. On the other hand, if your sample contains other nucleic acids that are not fully complementary to the strands that are immobilized here, then even if you get some partial matching, partial complementarity, this is going to be very, very unstable because you don't have enough hydrogen bonds. And so when you wash, this is going to come off. And what you're left with is whatever DNA or RNA sequence has full complementarity or the most complementarity to your probe, right? So basically you have some specificity by being able to have the most complementarity toward your target analyte. Another kind of setup that you can utilize is the sandwich hybridization method, similar to the sandwich immunoassay that we described for antibodies. But in this particular case, you're going to have a capture antibody that is attached to your transducing surface. We then are going to add our target nucleic acid, but in this case, there's not full complementarity. There's just going to be like a partial complementarity to that capture probe. And then what we do is that we add a signaling probe, which basically is another nucleic acid sequence that is already labeled in some way, let's say with a fluorescent molecule. And that one is partially complementary to the overhang of the target. This kind of tail is going to be known as the overhang, whatever part has not yet been hybridized to the capture probe. And so once they do hybridize through base pairing, then now you have your whole sandwich system, which is now fluorescently labeled, and you can read it utilizing fluorescence spectroscopy, for example. Another platform or another method is hybridization. Via hybridization is a microarray system, and this is similar to the microarrays that we mentioned for proteins, where basically you would take your cell or tissue sample, you would extract, in this case, RNA or DNA as opposed to proteins. Once you have this is going to be a mixture of all of the DNA or RNA that your sample had, right? No sequence specificity yet. You then are going to amplify it so that you have more quantity of it, and you're going to label it typically with a fluorescent molecule. But right now what you have is still a mixture of all of the DNA sequences or RNA sequences that are found in your original cell or tissue, and they're all labeled with the same fluorophore. The way that you have some specificity is now by adding it to a pre-prepared microarray in which we have selectively placed nucleic acids that are different So this is one type of nucleic acid. On this spot, we have a second type of nucleic acid, and so on and so forth. They have different sequences, and therefore, they're going to attach to different nucleic acids from our sample. Of course, that requires that we have some prior knowledge of what it is that we're looking for. So if we know the sequence of the target, then we can put a good probe, a good capture probe, to enable them to hybridize and be able to basically capture them and immobilize them onto our array. After that, of course, you are going to be detecting the fluorescence from the actual sample here. And the spots where you do get fluorescence, it means that you have the complement to your capture probe, basically. And from there, you can then collect and analyze the sample and determine, what nucleic acid sequences were found, etc., etc. All right, a completely separate way of detecting binding of nucleic acids to our nucleic acid biosensor is by the use of intercalation dyes. So an intercalating dye is basically a small molecule, typically a planar polycyclic molecule that has a positive charge, and that has basically, because of its structure, it has the ability to embed itself between, how can I explain this? Between what's known as the groups or spaces that are found in between hybridized nucleic acids. So for example, here I'm showing natural DNA, which is double-stranded, hybridized, right? And as it folds, it's going to have this kind of holes, these kind of cavities that are going to be just the perfect fit for these kind of molecules that have this kind of structure. Those molecules, which are now represented here in black, are going to embed themselves in between that hybridized DNA or RNA. And typically what happens is that when they get embedded in this structure, they're going to change their fluorescent properties sometimes. And so we can use that as a way to detect. So now, well, here is just kind of a demonstration of how they would embed. These are actually two molecules that do this. One of them is interesting, is actually a chemotherapeutic agent. The difference here, there's two of them, in that they have the same structure except for the R group. In the case of daunomycin, it is a hydrogen. In the case of doxorubicin, it is a hydroxyl group. And then here on this right-hand side is showing you kind of how those molecules would embed themselves into the, I guess it says in between two base pairs, and it just has the right configuration to be able to stack within the bases of the nucleic acid. Now on the right hand side, I'm showing you how are we going to use this for biosensing. So one of the scenarios is that initially you have your probe, which is a nucleic acid, which is in a hairpin configuration perhaps, or a double-stranded configuration, and you have embedded already the dye within. And when it is embedded, when it is intercalated, it has strong fluorescence for this particular dye. Whenever the target comes, the target could be another nucleic acid that is complementary to this, or it could be a molecule that is known to bind to that nucleic acid. Then what's going to happen is that you're going to basically displace that intercalation dye, and that intercalation dye is now going to be in water in the buffer system and not stabilized by the nucleic acid. And because of that, its fluorescence is going to change. In this particular case, it decreases. And that's the case actually for many molecules. And so obviously by looking at the change in fluorescence, you would be able to know that the target has arrived. The target has displaced your intercalation dye. I'll go ahead and mention, for those of you who might work with cells in the future, DAPI, which is a molecule that is typically utilized to label the nucleus of cells, is one of these intercalating molecules. And basically, you guys probably will be familiar with that. Basically, when it gets into the DNA of your nucleus, it fluoresces intensely. And so even if you don't wash off any of the DAPI that remains surrounding your cells, you will still see significantly higher fluorescence on the nucleus, because of this, because when it is embedded here, it actually fluoresces a lot. So it's a really good dye for microscopy imaging purposes, of course, but also for biosensing. All right, another configuration is if you have your probe and your target. So the target is the nucleic acid that in this case we're trying to detect. And so when they are complementary, they're going to bind, and this is our binding event. And then we can add our intercalation dye, which would only attach if you have the double-stranded configuration. So if there's no double-stranded, like if the binding didn't occur and you still just had your probe, the intercalation dye would not be able to interact just with the probe. It would have to interact only with the hybrid system. And so because of that, now we have fluorescence happening. Whereas if we don't have the complementary or the double-stranded configuration, then we're not going to have fluorescence. I hope that I explained that somewhat clear. Okay, so the previous slides were talking about how do we detect binding to a nucleic acid. Our target was a nucleic acid, but in some cases we want to detect other things, for example, a protein, or we want to detect a cell. And so for that case, we can still use nucleic acids, but it's going to be in a different configuration. Specifically, we're going to be utilizing aptamers. So an aptamer is a synthetic nucleic acid that has been selected because of its affinity for the target. And that target could be a small molecule, it could be a protein, or it could even be a cell. The way that aptamers are developed are by utilizing this systematic evolution of ligands by exponential enrichment or SELEX process. And for that, we're actually going to be beginning right here. This is going to be our first step. So you're going to begin with a single-stranded DNA library. What that means is that somebody has prepared a system that has maybe 50 nucleotides or something like that have different random sequence. And they are so multiple, many, like hundreds of these individual DNA strands that have different sequences, except that all of them are going to have the same primers on the end to enable us to do amplification later on. But the middle part of them, which is the important part, is going to be just random sequences. And so basically what it means is that we have a bunch of different nucleic acid strands with different sequences that nobody knows really what their sequence is yet. It's just random. From there, what we're going to do is that we're going to combine that mixture of nucleic acids together with our target. And again, that target could be a cell, or it could be a small molecule, or it could be a protein. We're going to allow them to interact. And after some time, some of those nucleic acids, just by chance, just because of their structure, because of their fit, some of them are going to bound to the target, to some epitope on that target. And the ones that do not bind are going to be washed off. We then collect the ones that do bind, and that's going to be basically the extraction. And let's go ahead and skip this right now, but we're going to remove, actually no, we cannot skip it. We're going to remove, we have already removed the ones that didn't bind, and we're going to take the ones that did bind. And what we're going to do now is a negative selection step in which we're going to try to remove the ones that bind to some competing molecules. So let's say that I'm trying to detect glucose. And I'm trying to develop an aptamer for glucose, which is going to be my target. Well, I wanted to bind to only glucose and not to another sugar. So I'm going to use another sugar here as a control to be able to remove any kind of nucleic acid strand that has binding to that interfering molecule. Or for example, if our target is a liver cancer cell, then I might use a red blood cell or something like that as a negative control to remove those that also bind to that control, to the thing that I don't want to bind. Anyways, from there, we now have the extracted sequences that are put together with the control. This is the negative selection step, where we're going to remove all those sequences that bind to the thing that we don't want it to bind. And what we're left with is some nucleic acid strands that have affinity to the target, but that don't have affinity to the control. We can then do amplification of them utilizing PCR or polymerase chain reaction. And then we can start the cycle all over again with now a reduced number of sequences that we already know have some affinity to the target. We again put it together with the target to do a positive selection. We do another negative selection. We re-amplify, and now we start again with a new, with another library that is even more selective for a target. And typically this process is repeated maybe 5 to up to 15 times until you are left with a small number of nucleic acid sequences that you know have really high affinity to your target. And at that point, what you can do is that you can actually sequence, figure out what the sequence is, and that's a process known as sequencing, to know what is the letter sequence of A, T, G, C, whatever. And then you can do further characterization to learn more about the affinity and the sensitivity of that aptamer for your target. And again, basically, since we're starting with a random pool of nucleic acids, what we're doing is basically saying that by chance, one of those nucleic acids is going to fold into the right tertiary structure to be able to fit within an epitope on our target, any kind of epitope. Now, keep in mind that the target could be a small molecule that might be smaller than the aptamer, in which case that small molecule will actually fit within the aptamer, or it could be a large thing like a cell, in which case the nucleic acid is actually going to fit in some kind of pocket or something like that of the cell itself, like on a protein or a sugar component on the surface of the cell. All right, one last thing that I wanted to mention before I move on from this slide is that aptamers were first developed in the 1990s. There were two main papers that came out in that same year mentioning this kind of technology. One of them was by Dr. Ellington Andrew Ellington at the University of Texas at Austin. And so he's a really cool guy that you might want to read papers about. All right, so as we mentioned, these aptamers are going to have the right tertiary structure to be able to bind to a target. And in this particular case, I'm showing you just two examples. This is one aptamer that binds to theophylline, which is a small molecule. Of course, the aptamer is much bigger than the molecule itself, since this is basically very similar to one of our nucleotide bases. But it just has the right fit and has been studied to be able to bind theophylline with high affinity. And then on the right hand side, I'm showing an aptamer that binds to a protein. So the aptamer is actually the one that I am kind of tracing right now. It has a hairpin structure, and you can see that it actually is known to fit within this kind of cavity in the middle of the protein. Right here, this is my protein, one side of it and the other side of it, and the aptamer just fits right in. All right, so what are the benefits of aptamers? First of all, they have similar affinity to the targets, just like antibodies. So you know, they both have the potential to act as biorecognition molecules. The next benefit is that they are easy to synthesize through that solid phase synthesis, and because of that, they're also really easy to modify with labels or attachment chemistry with site specificity. So you will know exactly where that fluorescent molecule has been placed. You will know exactly where that functional group is being placed to enable you to immobilize it to the surface. That's different from an antibody, because in antibodies, the protein itself had so many functional groups in every single amino acid that you had very little control of where the chemistry occurred. They are easier to immobilize, when one of the reasons being that you have control over where the attachment chemistry is at, and another reason is that they are more stable, and so you can do the chemistry with a little bit more ease. They are less expensive in general, unless you start putting a lot of labels and attachment chemistries, in which case they can be really expensive. They are more stable than proteins for sure, even though we still have to be careful because, as I mentioned, they are labile to the action of nucleases. And they are easy to regenerate. As I mentioned, you can denature your nucleic acid, and after you remove the input, whether that was temperature or changes in pH or something like that, the aptamer can refold again once you reach back to the original conditions. Now, the disadvantage of aptamers is that they can be degraded by nucleases, as I mentioned. However, the stability of them can be improved by chemical modification, as I mentioned in one of the early slides. Now, in terms of the transduction methods for nucleic acid-based biosensors, there's going to be two main ones. One of them is optical approaches, and the second transduction method is going to be electrochemical approaches. In these few slides, I'm going to focus first on the optical approaches. And so for optical detection of nucleic acid biosensors, we're going to be utilizing three main approaches. One of them is fluorescence quenching, in which we're going to be utilizing a fluorophore or fluorescent molecule that is going to be placed in close proximity to a quencher. That quencher is going to be a small molecule typically, or a metallic nanoparticle that has the ability to quench the fluorescence of this fluorophore. And so here the distance between these two is actually going to control whether or not fluorescence occur. If they are in close proximity, the fluorescence is quenched. If they are far apart, then the fluorescence can be increased. And of course, you will see in a little bit a couple of architectures for a biosensor that is going to enable changes in the distance between these two. The second or the third type, I should say, of approaches for transduction is going to be fluorescence, resonance, and energy transfer, or FRET. In FRET, and I should also mention that sometimes the F stands for Forster, resonance energy transfer, and you'll find it with both names. Really, the proper way would have been to say Forster, and I apologize that I just remembered about that. But basically, FRET is going to be a technique that is based on the energy transfer between a donor chromophore in its excited state in an acceptor chromophore. What that means is that we're going to have a fluorescent molecule, which is going to be our donor, that is going to be excited by irradiation of light that it is able to absorb. When it is in the excited state, that molecule is going to be able to transfer the energy absorbed to a acceptor chromophore. But for that to be able to happen, there has to be two requirements. One of the requirements is that they are in close proximity. The donor and the acceptor molecules need to be in close proximity, typically less than 10 nanometers apart. And the reason for that is that the energy transfer efficiency is going to be inversely proportional to the sixth power of the distance between them, according to the equation down here. And so if they are too far apart, no energy transfer can occur. And that's shown graphically here on part B of this figure. Basically, we're going to have this is the donor molecule. It gets excited by a specific wavelength of light that it absorbs, typically with fluoresce at a little bit of a longer wavelength. And if these two molecules, this is our donor and this is our acceptor, if they are far away, more than 10 nanometers apart, then there's no energy transfer between them. On the other hand, if they are close together, less than 10 nanometers apart from one another, when we excite this one, it is excited to, the electrons are excited to an excited state, but then it is able to transfer that energy to the acceptor molecule. And now that acceptor molecule is actually going to fluoresce. So what you're going to end up having is that you're going to see a change in the wavelength of fluorescence, because now you don't see the fluorescence from the donor molecule, but instead you're going to be seeing the fluorescence from the acceptor molecule. So you'll see a clear difference in the wavelengths of emission, and that's a direct indication that the proximity of those two molecules has occurred. And again, I'm going to show you in a little bit how do we set up our biosensor to change the distance between these things to indicate binding to the target molecule. Now, the other requirement for FRET to occur is that the absorption spectrum of the acceptor molecule has to overlap with the emission spectrum of the donor. What that means is that the energy that has been accepted by that acceptor needs to be transferable. The energy that has been already absorbed by what we know as the donor has to be transferable to the acceptor. And the only way to have that happen is if the two overlap. And that's actually shown in part A. So let's label this A. This is label, label this as part B from the figure on the right hand side. So anyways, for part A, what it means is that the emission, the fluorescence spectrum of the donor needs to overlap with the acceptor excitation or absorption. If there's no overlap, then no FRET is going to occur. While here, if you do have spectral overlap, then the FRET process can take place. Now, I should mention that what you're seeing here, these little humps, this is wavelength on the x-axis versus intensity of absorption or fluorescence on the y-axis. So what we're seeing is that if the wavelengths don't match, then no FRET could occur. But if the wavelengths do overlap, at least to a certain extent, then you can have the transfer take place, because basically the amount of energy that has been absorbed and that it is in the donor, it's enough to be able to excite the acceptor. That's basically what it entails. All right, so in general, if you have FRET happening, what's going to happen is that you're going to have a change in the wavelength of emission of your fluorescence, even though you are exciting with the same excitation light for the two setups. But, and if you're looking at quenching, what's going to happen is you're going to have a change in the intensity of fluorescence of that one fluorophore. If they are close together, you're going to have quenching, so lower fluorescence, while if they are far apart, then you're going to have higher fluorescence for that fluorophore. All right, so how do we set up our architecture for the biosensor to be able to take advantage of FRET or of quenching? These are some of the probes, some of the architectures that have been utilized, particularly for the detection of nucleic acids. So this is the target nucleic acid that we're trying to detect, the one that is kind of in green. The blue things are going to be our biosensor. Notice that in the first two sets, these are known as binary probes because we have two different strands that are acting as our biosensor. Those two strands have complementarity to different parts of the nucleic acid that we're trying to detect. So when we and the other thing that you should note is that in part A, what I have here is a fluorescent molecule, our fluorophore, and then the black one represents the quencher. So when these two things come together, if they come like this together because of the sequence complementarity, now they are so close to one another that the fluorescence is going to be quenched, versus in solution, when the target is not present, you're going to have high fluorescence. And of course, what this means is that as the concentration of the target analyte increases, you're going to have lower and lower fluorescence. So it's a turn off kind of biosensor. In the second one, in B, what we have is a fluorophore number 1, which is going to be our donor, and a fluorophore number 2, which is going to be the acceptor for FRET. And so in this particular case, in the absence of the target, these two are not next to one another because they're just kind of freely swimming in the solution. And because of that, we're only going to be seeing the fluorescence from the donor molecule. But once they are in the presence of the target, they are going to hybridize according to complementarity of the nucleotide bases. Then now they're brought together into close proximity. And because of that, even though we are exciting this donor molecule, it is going to transfer its energy to the acceptor, and we're going to see fluorescence from the other molecule. The bottom two are molecular beacons, which are basically hairpin structures that contain a loop that can recognize the target molecule, and a stem that is unstable enough to be able to dissociate when the target binds. And we have labels on the 3' and the 5' ends. The labels could be, again, fluorophore and quencher, or fluorophore number 1 and fluorophore number 2, meaning donor and an acceptor, donor and acceptor, sorry. In this particular case, initially they are actually in the quench state, but once they actually bind to the target, they are far away from one another and the fluorescence increases. On the other hand, here we're going to have initial fret occurring because they are in close proximity, and so we have fluorescence coming out of the acceptor, but when the probe or the beacon binds to our target, they are far away, and you're going to be seeing the fluorescence from the donor only. All right, now on this slide, I have the architectures that are utilized for aptamer probes. So the difference here is that our target analyte is going to be a molecule other than a nucleic acid. And so we're not utilizing complementarity in terms of base pairing between the target and the biosensor, but instead some kind of affinity interaction, which is what we said that the aptamer could do. Now the configurations are multiple. One of them is to utilize the molecular beacon that I showed in the previous slide, but in this case the binding of the target to the biosensor is going to result in different kind of folding, basically some kind of folding around our target that is going to change the distance between the quencher and fluorophore in this particular case, or between the fret pairs, the two fluorophores if you have fret. The second configuration is one where the initial aptamer actually does not have kind of a hairpin structure, but instead is more like a random coil. But when it binds to the target, it actually folds in such a way that it decreases the distance between the two probes, leading to either quenching or fret to happen. The next one is known as a structure switching aptamer duplex system, in which you have the, you have actually, it's a duplex, but it has actually 3 nucleic acid strands. It has this guy and this guy, which are partially complementary to a long strand. That long strand is going to be our aptamer, but is going to have an extension. So the right-hand side is the aptamer. Something like up to here is typically the aptamer. So that basically it is bound to this partially complementary strand by a few nucleotides. So it's unstable enough to be able to displace this. When the target comes, this part is going to start unfolding or folding in a different way, and is going to end up displacing this guy to be able to bind with the target. The rest is just an extension to enable this stem to this part to bind, to hybridize. And so basically what ends up happening is that we displace one of the nucleic acid strands, leading to increase in the distance between our fluorophore and quencher, or in between the fret pairs. The next one that I'm showing is actually a split aptamer, in which we have this strand, which is part of the aptamer, and then this loop, which is part of the aptamer, and they are both in solution kind of separately. When we add the target, these strands are going to refold in such a way that they kind of make up the structure of the aptamer, of the full aptamer, and now you're going to be basically changing the distance in between these two. The next configuration is still a split aptamer, in which initially you didn't have any kind of tertiary structure like a hairpin structure as just shown here, but you had random coils with half of the aptamer being this and half of the aptamer being on the second strand. But again, when they come in contact with the target, they're going to wrap around that target in such a way that they come together, thereby decreasing the length or the distance in between the quencher and fluorophore, or in between the fret pairs. And then finally, we have this other configuration, which is similar to C, in which instead of having 2 strands that are partially complementary to the aptamer strand with its extension, what we have is the aptamer strand with its extension being hybridized to a single molecule. And now the labels are going to be one on the aptamer and one on the partially complementary strand. But this has to be designed in proper way such that it is unstable enough that when the target is present and the different folding occurs, that it would displace this, basically again increasing the distance in between the two probes. This next slide is showing a couple of additional examples in terms of fluorescent aptamer sensors. The structures are very similar. So here on the left, we have a hairpin structure with a fluorophore and a quencher, which is able to bind to either a binding protein or a larger protein like thrombin, for example. And as we mentioned, basically there's going to be a change in the configuration of that aptamer architecture, where it's going to change the distance in between the fluorophore and the quencher. And what that shape is going to be very different depending on the aptamer. On the right hand side, I have actually three different configurations, or actually two, I should say. Oh wait, no. I have three different configurations for other biosensors, but you can see it's kind of the same idea, except that in this one, initially you actually have no hybridization between these tails of the two strands, actually of the single strand, and they actually are brought together as opposed to made farther apart when the target comes. That's kind of what I was trying to get to. That here is showing you where they bring together the fluorophore and the quencher when the target comes. And again, that takes some designing to do. You have to kind of study the aptamer, its sequence and its structure to be able to figure out how can you set up your sensor to enable the changes in the distance between the two probes. I think I'll leave it at that on this slide. All right, the last two slides that I wanted to mention is actually some work that is happening in my lab. This is a project that I have been collaborating with Dr. Jennifer Irvin, who works on the preparation of conductive polymers. And so she's going to be providing the conductive polymer that is going to act as the transducer for electrochemical detection of biomolecules. And what I'm working on is the design of the probe, the aptamer probe, to be able to detect particular molecules of interest. And one of the things that, this is actually some preliminary data with optical detection, not electrochemical detection. But basically what we have is an aptamer, which is in blue. That aptamer has high affinity for the molecule adenosine, which is a small molecule. And the way that I configured it is that I use kind of the last approach from 2 slides ago, where I have 2 strands that are partially complementary to one another. One of them is short, right? But they do hybridize sufficiently, and their melting temperature is high enough that they would be in this kind of state at physiological temperature. And so because they are hybridized with one another, we have close proximity between the fluorophore and the quencher, and that means that we have no fluorescence in this state. But when the target, which is the adenosine molecule, comes, it basically ends up displacing the quencher strand, and in that case you have fluorescence development happening. To be able to design the right sequence of our quencher strand, what we did was actually to take the aptamer strand, which is shown right here. That aptamer strand, we then looked at different complementary strands, which are going to be the green strands, that are going to have different length. Notice that they are complementary in terms of the nucleotides to the aptamer strand, but they're going to have different lengths. The longer they are, the more stable they're going to be. And if they're too stable in terms of too many hydrogen bonds, then the adenosine is not going to be able to displace it. But if they are too short, then the melting temperature is going to be too low, meaning that the hybridization can be disrupted very easily, and we might get false positives, where in the absence of adenosine, this strand is going to come off from the aptamer strand. And so we investigated all of these systems, and we found that number 4 was actually the one that enabled us to be able to detect adenosine at different concentrations. And that's the data that I'm showing here on the right-hand side. It's basically concentration of adenosine versus fluorescence. And as you can see, as the concentration of adenosine increases, so does the fluorescence in a particular range. We then took that system 4, and that's actually the system that we are currently investigating in trying to put together an electrochemical biosensor. And here I'm showing the setup that we're utilizing to do the detection in the laboratory, where it's basically an electrochemical cell that has a working electrode, a counter electrode, and a reference electrode, very similar to what Isha had mentioned in the paper that she described. In our particular case, the working electrode is actually a glass slide that is modified on its surface with indium tin oxide to make it conductive. Now, on that glass slide with indium tin oxide, we actually electropolymerize our polymer, which is the conductive polymer. And that conductive polymer, this is one of the two that we're working with. But this particular one has these A-side groups that enable us to attach a functionalized nucleic acid that has an alkyne group utilizing copper catalyzed click chemistry. The second chemistry, or the second polymer that we're utilizing, is going to have carboxylic acids functionality, and then our aptamer is going to have amine groups to enable the formation of an amide bond between the two. But anyways, we utilize the setup to be able to polymerize the conductive polymer onto the slide, and also to be able to look at the electrochemical response of that system upon interaction with the target molecule adenosine. We're still working on this, so I don't really have any particular data for this just yet, but hopefully we will in the future. And we will also be working with aptamers other than the adenosine aptamer. We're particularly looking at being able to detect cancer biomarkers. So with that, I will leave you guys. This is the end of the lecture notes for this class. I hope that you guys learned a ton, and I hope to see you guys in the future. Bye-bye.